Okay, good, excellent. We can get started. So today we will complete the discussion about the basic building blocks of the life, and we will start to talk about more about the mathematics foundations of the diffusion and the physical meanings of the diffusion. We will do the derivations in the next few lectures. This is the most fundamental process in the cell body. Okay, so regarding to the logistics, everything else is the same as before, and I only have one thing to update. That is, some of you request to have some examples of midterm exam number 1, right? So I put some example questions on the canvas now, so you can use that, and the exam will cover 10 questions, and it will be the same style. Okay, so then you can get yourself familiar with what kind of questions we will ask during the exam. Okay, homework question will be more involved. Those questions, because you have more time to do that, so you can take the time to use the references. But the exam, you need to do it quicker. So it's mostly to ask conceptual questions. Okay, so as long as you understand the material, then you should be no problem. And it is open book and open notes. Okay, good. So that's all for the logistics, and feel free to continue to send to me the survey. Okay, so many of you actually send me the survey after the class. That's good. Some of you have not sent it to me, but if you have any opinions, please feel free to do so. Okay, you can send me survey, or if you have a lot of things to say, we can schedule a time to talk in the office hour as well. Okay, so I hope to use this way to have 2 ways communication among us, so that we can improve the class and help and enhance your learning experience. Okay, good. So if no other questions, let's start today's lecture. So in last lecture, we talked about the basic properties and functions of the sugar. And then we also started to talk about the second types of macromolecule, that is the protein, right? So we talk about the protein, that it is the most, it accounts for the most of the dry weight in the cell, as well as in the bacteria, right? So that is more than half of the macromolecule in the cell, because protein is so important to perform different functions in the cell. And we talked about those functions in last class. And then here is a cartoon animation, shows one of the protein, one types of protein, that is a motor protein, that it can walk around the microtubule. It serve like the bus, right? Serve the bus and to carry the cargoes on the highway. Yeah. So this means the protein is indeed really important. And if we look into single monomer of the protein, we will find that all the monomers actually share the same structure. Mostly share the most same structure, except only one thing is different between different monomers. That is a functional group, called R group. Okay, and then those monomers are called amino acid. In total, so far, people have found 20 types of amino acid. Okay, all those amino acid has a central carbon atom, and then four side groups. Amino group are called the N terminals, because this is the starting point of the amino acid chain. Okay, and then end at the carboxyl terminus or C terminus, because this is the end of any protein. And then it has a simple but important hydrogen side chain and a distinct functional side chain, or called R group. This R group determines the property of the amino acid. Okay, all other parts of amino acid are the same for all of them. And then we talked about the different. We talked about one type of categorization of the amino acid based on the charge. If we based on charge, then based on the polarity of the side chain, then we can categorize all 20 amino acid into 4 groups: non-polar side chain, polar side chain. Both of them actually are the amino acids that have overall neutron charge. Okay, and then also the electric charged side chain. So here is the negative charged chain that actually those amino acid is more acid. And then the positive charged side chain, those only 3 amino acids belong to this category. This is more basic. Okay. Okay. That's the table of the all 20 amino acid group. The most common grouping method is based on the charge. And here it is the corresponding genetic code for the amino acid. Now, do you guys hear me?
Yep.
Yes.
Okay, very good. Now, this table is very important. It captures the 2 languages we talked about: the genetic code, nucleic acid, and protein code, amino acid. Okay, it takes both. Now, here you see 5' and 3'. This actually means the direction in the gene. So when we talk about the next building block about the nucleic acid, you will see what is 5' and what is 3'. And remember, for the protein, it's not 5' and 3'. Protein is N terminals to C terminals. It's from N terminals to C terminals. Okay, for the protein. But for the gene, it's 5' to 3'. Now, we have this actually converted to RNA already, but you have U here. Okay, no T. DNA has T, RNA has U. So this actually means from the 5' on for the RNA, you pick up RNA has only 4 subunits. So you pick up any one, and then that one goes to another. The other one has 4 different possibilities. So this is radially, you look at it radially from the center and radially outwards. Okay. So first you pick any amino. By the way, each amino acid here actually is determined by a corresponding genetic code that consists of 3 nucleic acids. Okay, so 3 nucleic acid contains the code for the 1 amino acid. And if it's 3, and every nucleic acid has 4 chances, right? 4 chances. So 4 by 4 by 4, 3 times together. So that's the 4 by 4, 16, 4, and then 64 possibilities, right? So from the center on, you choose 1 nucleic acid in the RNA, another, and then you have 4 chances to pick the second position, and then you choose any one, and then you get another 4 chances, right? So, and then from there, it corresponding to 1 amino acid, okay? 1 amino acid. So you can see here, actually, it has redundancy. From this one up, GAG, it actually corresponding to E, okay? Corresponding to the E. And this E is glutamate, okay? And then GAG also, GAA and GAG both corresponding to this, okay? So this happens a lot. People have actually decoded all those correspondence. So any 3, any 3 corresponding to 1. And then sometimes actually it's a 4. GUG, GUA, GUC, GUU, all corresponding to the V. Okay. And some of them actually do not corresponding to anything. so far people have not find it. Okay. Now, this from here you can see that the any one of these three nucleic acid code together, that's called a codon. These three acids called codon. Any one codon corresponding to one amino acid, but any amino acid can corresponding to many different codons, right? So this is not a one-to-one correspondence. So that's why in previous class, one of you actually asked a very good. I asked that question. I asked in the central dogma why the protein cannot go back to the RNA, why the RNA can go back to the DNA, but the protein cannot go to RNA. So one of you told me some answers, and then I think that answer is very good, but not complete. The complete reason to make it complete, another reason should be considered is if you want to convert the protein into the RNA, it has many possibilities. So this one can correspond to many of them. So then you need a mechanism to tell the cell which one you need to correspond to, which one it is originally coming from in the protein, which codon originally gives you this amino acid. Then if you find that mechanism, then you can go back. Okay. So far nobody have found the mechanism that mechanism yet. Okay. So when those amino acids combine together, they actually also perform the condensation reaction that we talked about in last class. So they can take out one oxygen, one OH unit combined together, and then generate a molecule, a water molecule, and then form the covalent bond to connect two amino acids together. That covalent bond is called a peptide bond. And then when they combine together, they actually always is the N terminals connected to the C terminals, always connected to this one. Okay. Here, by the way, Daniel last time asked me the question about the structure. Here, they actually is the sense structure. Okay, this is a sense structure. In last time is also sense structure, just in terms of the writing on the PowerPoint slides, it looks different, but it's a sense structure. Okay. Now, Daniel, you hear me?
Yeah.
Yeah. Okay. So now it's clear, right?
Yeah.
Excellent. Excellent. Keep asking those questions. That's very good. Excellent. Good. So the protein, once they form those long polymer, once they form those polymer, then they will not stay in the linear. Remember the table I showed you, the charge table. So some of them are positive, some are negative. So then they actually will attract each other and repel each other. So they will not stay in the linear polymer. Then they will form a 3D structure. Okay, so that is called a 3D conformation. Those 3D conformation is very important to account for the function of the protein. And if there are during the translation process, if any amino acid get a wrong sequence, then the final 3D conformation will be different. Then the function will be different. That actually causes a lot of diseases. Okay, such as, just name a few, such as Alzheimer's disease, right? Alzheimer's. So people start to forget, lose the memory now, right? And then the cardiac fibrosis, and then the Parkinson's disease, okay? And then medical disease, right? Epidemics, right? So that all of them actually is caused by the errors in the protein folding, special proteins, they're folding, okay? And then they can accumulate, so they accumulate toxic, insoluble gunk, okay? So in the Alzheimer's, here is actually show two pictures for the healthy brain and the Alzheimer's brain. You can see the comparison. So the healthy brain, the neuron actually go very straightforward, connected to each other. But for the neuron in the Alzheimer's disease patient's brain, there are many gunks, plaques generated in the brain, and then neuron cannot work that easily. And then the neuron will change their shape because this environment is not that present. So then neuron changes shape and then lose many functions. Okay, so these actually all are coming from the protein folding mistakes. Okay, and that's why the conformation of the protein determines the function, and then the function continue the proper function in the neuron actually can help neuron to generate the protein with the correct conformation. So they actually go in a positive loop. Okay, so we do not want to perturb this loop. So protein, as I said, that they form the 3D conformation. This actually based on the size of the 3D conformation, people can categorize the protein into 4 different categories. It's called primary, secondary, tertiary, and the number 4s. Okay, so for the first one, primary category, they are actually is the linear sequence of amino acid from N to C. Okay, so they are also called beads on a string. Beads actually is the amino acid atom. String actually is that peptide bond. So they are connected together. So this is beads. This is beads, and this is the string. Okay? Beads on a string. So this is the primary structure, and we can see those primary structure actually is relying on the covalent bond, right? Very, very strong covalent bond, peptide bond. And then those long polymer will adhere to each other, attract each other, and repel each other, and form the secondary structure. This is one first level of folding. So you can see here two types of actually see this one. This is actually secondary structure. Secondary structure. And then here it shows 2 types of structure. One type of structure is actually due to the attraction, the oxygen and the hydrogen in the amino acid chain, they attract each other. And then they form the hydrogen bond. For this one, they form a helix structure. This is called alpha helix. And they form this structure because of the hydrogen bond in between, okay, across the amino acids, okay. And then this is the one type of structure. Another type of structure actually is this linear one here. So this actually they more form a sheet, and then aligned polymer and another aligned polymer, they parallel to each other, and then if many of them look at it together, you will see it's very, very similar to a sheet. Okay, this is called a beta platted sheet. Okay, these two actually right now I'm showing here is the cartoon. In fact, we also have seen their structure in the electron microscope in the nature. If you take the protein right now using the electron microscope, you can see now very, very clear the alpha helix and the beta sheets. Okay, so this is truly existing in the nature. Okay, and then yeah, for this kind of a secondary structure, they are mostly the peptide backbone is polar. Okay, is polar. Okay, so that they can adhere, attract to each other. Right? If it's non-polar, then they will not have electrostatic interactions. Right? So this secondary structure, they are mostly consisting of hydrogen bond. So then you can see hydrogen bond is you can see that this hydrogen bond actually is weaker than the covalent bond, right? If we apply force to this polymer, those hydrogen bond can break. We can straighten this alpha helix, but we cannot break it. Because break the polymer takes more energy to break the covalent bond, right? And then,
Doctor,
yeah, please.
What ensures that the protein folds in that way?
Say one more time. When?
what causes the protein to fold that way? Because you mentioned that like Parkinson's disease and mad cow disease, they are the result of like protein folding incorrectly.
Yeah.
So
Excellent.
what system ensures that the protein is folded correctly?
Excellent question. Protein folded correctly based on their sequence. Based on a sequence. Right now all the protein in the nature, they are after many billions of years of evolution. So the bad one already get eliminated. The remaining one is good one. So those are good one. We think they are correct and they maintain our function. And if the good one during the cell division, if there are some mutation happens, genetic mutation happens, sometimes it happens, such as when the cell divide, there is a UV light turn on, accident UV light turn on, and then the DNA can mutate. And then once it's mutated, then in the 3 amino acid codon, then one of them is wrong. And then it will corresponding, if it corresponding to protein corresponding to amino acid A, now it may be correspond the ribosome will translate it into amino acid B. Then it's a different amino acid. That different amino acid may have different polarity and electrical charge. Then that one will adhere to the neighboring amino acid in different way, right? Then the folding conformation is not correct, right? That will cause trouble. You hear me?
Yeah.
Yeah. Very good question. Yeah. So they actually in right now many people is designing the new protein. So when they have the software, it's called Lawson software. So that software you can put in the amino acid sequence there, and then the software can do this for you. They can find out the hydrogen bond in the computer, and then form the 3D structure on the screen. Then you can test, you can check whether that is the structure you need or not. Okay, so there people actually you can do it in 2 different ways. One is you can get the 2 ways. You can get the sequence done, and then use the sequence to predict the 3D structure. Or you can go reverse. If you want a certain 3D structure in your experiment, then you can go reverse, design the corresponding sequence that can go to that 3D structure. Either way can be done. Okay. Good question. Yeah. So the secondary structure of the protein actually is very important. For example, the hair protein, keratin is actually in our hair is very rich in the alpha helix structure. Okay, and then these hairs, our hair actually can stretch because it's easy to break the hydrogen bond that sustaining those alpha helix. Okay, and yeah, there are computer modeled secondary structures. So this is the computer model, the cartoon showing the alpha helix, and then also some of them actually combine with the beta sheets. Okay? If we go even further, we put those secondary structure even further. So here actually shows all 4 kinds of structure. Primary, we have seen this is the sequence, and the secondary it contains alpha and beta. And then if you go even further, tie them together, you will form the 3rd structure, tertiary structure. Okay, this fold the helix and the strand into a domain. So this is like 3rd structure, and then 3rd structure, you tie them together, you get a 4th structure, quantitative structure. Okay, so this is the biological units. This actually is close to the final protein. It's a final protein. Okay. Okay, so that's all for the second building block. Now let's go to the 3rd building block. That is the nucleic acid. Do everybody
Dr. Tong, I've got a quick question.
Please.
So I've seen a good amount of this before in Bio 1, but I've always been curious, how are the diagrams of the structures created? Like the one you have in the bottom left with the alpha helices and pleated sheets.
How this is created?
Yeah, how is the diagram itself created? Because I thought it had something to do with a lot of computational
Yeah,
power and probability.
yeah. Create of this will not take too much. These are created in the computer, but the creation is taking too not taking too much computer power. Okay, but if you do the simulation about when how the function of those protein change in different conditions, that takes a lot of computational power. So usually people only simulate for less than a millisecond. Less than millisecond. Within 1 millisecond, what's the function of this protein in that specific condition? Because this takes a lot of power. And the reason is the protein actually is very dynamic. They are keep interacting with the water molecule and with themselves. And that speed is very, very fast. So if you want to know the exact sequence of the process, you want to divide that 1 millisecond into thousand or millions times delta t. So that, and then each step you need to count all the coordinates of all the molecules. So you will need both a lot of frames as well as a lot of coordinate to be taken into account. So that takes a lot of computational power.
Right, interesting. Doesn't our chemistry, I don't know if this is something you're familiar with, but I believe our chemistry department does stuff with Hypergator, the supercomputer, to do things like that with molecule modeling?
Yeah, you can do that. Yes. Mm-hmm.
Okay.
There are very good molecular dynamics softwares that can perform those kind of calculation. The basic law how this molecule work is known. That theoretically is known, and the quantum mechanics is there to help. But to implement that into those large molecule, large molecule, that is very power intensive. Yeah. Good.
Right. All right. Thank you.
Excellent. Excellent. Good. So the third macromolecule is nucleic acid. So nucleic acid, we only talk about the two types. One is DNA, one is RNA. They both act as the information storage molecule. Okay, they have more other functions, but they definitely have the information storage functions. Okay, for the DNA, the name actually is coming from deoxyribonucleic acid. Remember, we talk about the deoxyribose, right? We talked about that. That actually is this one. Okay, this one, similar to this one. Okay, and then the ribosome nucleic acid. Okay? That is RNA. Their monomer is called a nucleotide, or sometimes we write as N.A. Okay, the basic structure, any nucleic acid, basic structure is they have three parts. One is the sugar, the ribose, okay, the sugar, the green one, and then has a base in purple, and then has a phosphate that is connected by the three phosphor atoms. Okay. And remember, we talk about the ATP, adenyl triphosphorate. So adenyl, so this will be the A adenyl, and then triphosphorate. So ATP is actually one of the nucleic acids. Now, if we look at the base, that purple base, we will find that there in the nature we actually have find 5 types of nucleotide base. There are pyrimid and purine, right? Pyrimid, you have the uracil, CU, and cytosine, C, use the first alphabet to represent it. And then thymine, T. And then for the purine, you have adenine. Remember the adenine triphosphate, ATP, adenine, right? It's adenine, A. And then guanine, G. We have the ATP, we also have GTP. GTP is another type of energy that works in the cell, mostly for the signaling, not for the just for the energy release, for the signal. Okay, so there are many GPCR in the cell that works with the guanine triphosphate. So this 5, actually, DNA has 4, and the RNA has 4. For the DNA, in the pyramid, for the DNA, it will have C and T. For the RNA, it will have U and C. So the RNA will not have T, it will have U. Any DNA, if they are transcribed into the RNA, the T region will be replaced by the U. Okay, and then they will share the same. They have the same purine. A and G are the same. Okay, so both DNA and RNA only have 4 chances for the nucleic acid bases. Now, here is the list of the base. Okay, A, G, C, U, T. Okay, and then this is the structure of any single nucleic acid. So for all the nucleotide in the DNA chain, they will have the same 5 carbon sugar and a phosphate group. So 5 carbon sugar is this one, and then phosphate group is this one. Okay, and all RNA chain also have the same 5 carbon sugar, but for the RNA will be the oxygen OH will be there. For DNA there will be no OH. Okay, and then they also have the phosphate group, but for the base, DNA and RNA will be different. They will show the you will choose this those different they choose from those different 5 units. Okay, and then the sequence of the gene actually comes from the can be written in terms of the linear sequence here. Here's one example showing the sequence of one DNA. This is one DNA because there's no U, right? This is T. Okay, so the difference between the DNA and RNA, they are mostly are 2 respects at the monomer level. Firstly is which nucleotide base are found in the nucleic acid. In the last slide, we showed 5 nucleotide. And then another one is which sugar is found in each nucleic acid. Okay. For the RNA, there actually is a ribose. It's using the sugar part. This is a green part. Is ribose, so it has OH. For the DNA, it's actually deoxide ribose. So OH becomes H. This is in the number 2 position. Okay. Now here is the more detailed comparison between the DNA and the RNA. So we can let's see. We talk about most of them. Here, most of them already. One is the different sugar base for H and OH for DNA and RNA respectively. Second one is DNA as a CT. The RNA has a CU. DNA as AG, and RNA also has AG. And then the monomer of this for DNA, the monomer of them are called deoxyribose nucleotide. For the RNA is a ribonucleotide. Okay, and now all 4 first comparisons are based on the monomer level. If you go to the polymer level, another difference is DNA is also always double strand, RNA is single strand. So this actually comes with a very interesting physics model. because the people have derived the linear chain model to derive the theoretical mechanical equation to calculate the mechanical rigidity of the DNA and RNA for those polymer chains, okay, in the polymer level. Yeah, and additional knowledge about the nucleotide is when they form when the DNA pair with each other, form the double helix, the A and T will form together. They have only two bonds, two hydrogen bonds. That two hydrogen bonds have about 2 kT energy. And the GC, they are actually has a GC combined together, they have 3 hydrogen bonds. That is much stronger, double the strength, 4 kT. Okay, and also there are many. Those are actually both of them actually is quite a weak bond, But the DNA usually has a very long chain. So many of those AT and GC bond form together, then they can be very strong. Overall is very strong. So then DNA is very stable. That protect the information contained in the DNA from being damaged by the harsh environment. Okay. If we want to read out the information from the DNA, then there are special enzymes that can be used to open those bonds, and then there are other type of enzymes that can start to read the information, and then take the free monomers to follow the sequence of the DNA, and then make a third polymer chain. Okay. Remember when we talked about the PCR in the previous class, talk about archaea living in the harsh environment. In fact, the PCR method can work is because people used a polymerizer that existing in the archaea that can sustain high temperature, and then they can do this job. They can split the DNA and then replicate the DNA, transcribe the DNA in very high temperature. In the 70 degrees C temperature, they can still do this job without any error. Okay, so then that can allow people to expand gene in the lab, in the wet lab. Okay, so those enzymes actually is existing in those enzymes and the molecular machines existing in the nature, and they are very useful. So here, as we see, for the single bond, single AT bond, single GC bond, there are 2 kT and 4 kT respectively. For any ATP molecule, if you use energy, they can provide 25 kT. So those enzymes actually take the energy here, take the energy from ATP. Every 1 ATP, they can take 25 kT energy, and then use that energy to compensate the breaking energy of this. Okay, so 25 kT can break 12, at least 12 AT bonds, right? 12 times 2 is 24, less than 25 kT. Right? So that's that's the quantitative description of the strength of the DNA.
What unit is KT in this context?
KT is the joule. So K is Boltzmann energy. This is a joule per Kelvin. So
right. Okay, yeah. I remember that now.
Yeah. And then if you look at this KT, they can also be converted into another form. That is 100 piconewton times nanometer. Okay, so Newton times meter is joule as well, right? So this is the same, and this actually can be this 1 ATP actually can supply the motor protein working for 1 or 2 steps. So those energies are very, very precisely defined and used by the molecular motors. Very good. So for the RNA, if we look at RNA, there are actually 3 types. There are many types, not only 3 types. These 3 are earliest discovered RNA. Right now, people find many more RNA existing in the cell, and all of them actually are indispensable. Those are the messenger RNA, transfer RNA, tRNA, and the ribose RNA, RR. Okay, so those messenger RNA actually is taking the information from the DNA inside the nucleus, and then diffuse out the nucleus, diffuse out from the nucleus into the cytoplasm, and then the tRNA will carry. tRNA actually carries three. You can see here three. Let me see. Messenger RNA comes here. tRNA carries the complementary base pair, and then one amino acid. This dot is amino acid. This dot, okay, one amino acid. So tRNA carries both 3 coat of the RNA and then 1 dot or one amino acid, and then join and then combine together. This is a ribosome. This is a dark one. Although it drawn very dim here, but this one is super important machine in the cell. It's super important. Now this tRNA comes here, but the messenger RNA will go through the ribosome. Once it enters the ribosome, the corresponding tRNA comes. It matches the 3 pair, so 3 subunit to the messenger RNA. Once it matches, then it will release the carried amino acid. This is 3 RNA base RNA unit, and this amino acid actually is following that diagram we just showed, that radially growing diagram, that from codon to amino acid. Okay, it follows that. It all follows that diagram, and then it comes to here paired together, then release 1 amino acid, then it goes away, and then this messenger RNA can move one more step further. In another step, then a new tRNA will come, match with that 3 RNA, and then release. If it's matched, then it release another amino acid. Then those amino acids is not just released; they actually will form the peptide bond, covalent bond, and then gradually release out. So there are beginning tRNA and ending tRNA, telling them when to stop. Once it's finished, then the whole amino acid chain will start to fold into 3D shape, and then it will traffic to different places of the cell to do the function. Okay. So here is actually a more magnified figure, shows that this messenger RNA is going. Okay, and then the tRNA carry the messenger RNA 3 coming, and then bond with that, match with that to do the checking. Once it's checked, then it release the amino acid, and then amino acid will form the peptide bond, and then gradually proceed to elongate the polymer chain. Okay, so those 3 are very important. RNA, and if you look at these 3, actually you will find something very interesting. That is, here RNA is not only used for transfer information, they also actually can have the function of catalytic function, because this ribosome RNA inside this DIN rich base, they actually is helping, is making up the ribosome, and it's helping this messenger RNA to do the work. Okay, so the RNA actually has a catalytic function. It's not like DNA. DNA is very stable. They do not do the catalytic function, do not promote other reaction. But RNA is different. They can do this, and this discovery. Once people realized this, they did more research, and that's actually that discovery actually lead to the Nobel Prize. And 2 scientists called Sidney Altman and Thomas Sicher. They actually, both of them are from Yale University. So Yale actually is doing great work in the RNA biology. One of my students actually went there for the summer summer research, and we have many collaborators there to do the RNA research in the application of the cancer and the brain science. So in terms of these two scientists win the Nobel Prize in 1989. Okay, and then here is the description coming from the Nobel Prize website. So they said the discovery of catalytic properties in RNA also gives us a new insight. So this is very important. Whenever you in the science, when you do things, you find a new insight that actually open a new field. Do you guys hear me?
Yeah.
Yes.
Yeah. Okay. Great. So this is the official evaluation of those two scientists' research. Okay. So they said they have given us a new insight into the way in which biological process once begin on the Earth. Okay. So the billions years ago on the Earth, actually the RNA is not only carry information but also has a catalytic property. That means it can help themselves to translate into the protein as well as regenerate into the DNA, right? So they can do this work in the two ways: in the center by staying in the center of the central dogma, right? Central position, C position, right? Now, researchers have wondered which were the first. So this is actually a long-standing scientific scientific mystery there. Researchers have wondered which were the first biological molecule. Which one was first biomolecule? How could life begin if the DNA molecule of the genetic code can only be reproduced and deciphered with the add? Decipher means decoded. Okay, the information be retrieved with the add of a protein enzyme. How can that be? If they are so secured, so conserved, okay? And then if the protein cannot carry those genetic information, and the protein can only be reproduced by means of genetic information from the DNA, right? Protein cannot carry those, cannot pass those information generation after generation, right? They cannot do that. So if that's the case, who is coming first, right? DNA and protein since are not the correct candidates, right? So then people ask, who comes first? Who is chicken? Who is egg? Okay. Then these two scientists actually is the person, they, based on the work we just discussed in the past slides, they realized very likely RNA actually not only carry information, they find that RNA, they find the missing link. Probably it was the RNA molecule that comes first, because this molecule has the properties needed by an original biomolecule. It is capable of being both genetic code and functional enzymes at one time and at the same time. Okay, so they are two-faced. The RNA is two-faced, so that they can do the work. They can be the earliest, the first molecule on the Earth. Okay, so that's their hypothesis. So the RNA can be catalytic. Okay. Now that's all for the nucleic acid part. We have many more to talk, but the time in the class is very limited.
I have a question on about the tRNA. So how does, like, I guess, is it like hydrogen bonding? How does the tRNA grab the amino acid? I'm assuming it has to grab it by the R group since it's specific by R group, but
yes.
Like what? Yeah, how does that work?
Yeah, so they actually have they do have bonds there, and once they connect to the once they are ready to release the amino acid, then the ribosome will help them. So they need to put a new energy inside to break that bond. That bond is not a hydrogen bond.
Covalent?
Covalent bond. Yeah.
Oh, okay.
Covalent.
So it's like between phosphate groups on the tRNA and the R group?
Yes.
Oh, okay.
The tRNA actually is very unique RNA. They actually can do both. They actually can, it's two-faced. They can connect both to the amino acid is connected to the amino acids terminal, and also has a certain region that can be easily grabbed by the ribosome to perform the bond rupture. Yeah, so it has a very special structure for TNA. It looks like, actually, it also looks very look like a T shape. Yeah, like this shape.
Yeah.
Okay, good. Excellent. So the last, oh, we don't have time now. So let's start in the next lecture now. So we will talk about the last category, the lipid. Okay, and then we can talk about the diffusion in the next lecture. Okay, great. Thank you, guys.
Thank you.
Excellent.
Thank you.
Thank you.
Thank you.